Hello, and welcome to Biotensegrity Tea Party number 16. This is season 2 of the Biotensegrity Party events, hosted by the Stephen M. Levin Biotensegrity Archive. Today we have our guest speakers here, Dr. Natalie Reznikov from McGill in Canada. Hi, Natalie. And Hila Tzipora Chase, who is doing her PhD in Montana. Hi, Hila, where are you? There you are. Hi. And the title for today's tea party is, If Bone is the Answer, What is the Question? Natalie and Hila will be sharing their biotensegrity-informed perspectives and research on bone architecture as they pursue this question, which was posed by Rik Huiskes, a legendary theorist in bone biomechanics, bioengineering, and prosthetic development. I'm Chris Morita Clancy, coming to you from the Coast Salish territories, just outside of Vancouver, Canada.
I'm Graham Starr from Robin Hood Country in the UK.
And I'm Susan Lowell de Solorzano from the United States colony of Washington, D.C. And it is our intention to promote and share the biotensegrity concept with as many people as possible and to include all of you in that mission. So we're glad you're here. The Biotensegrity Party is an all-volunteer production of the Levin Biotensegrity Archive, which is a nonprofit organization whose mission is education and building community and public awareness in the area of biotensegrity, and to foster and forward discovery and research and understanding in fields related to it, including science, health, and medicine. So.
Thank you, Susan. We like to open the Biotensegrity parties with a toast. And so we're going to invite Natalie to please give us our opening toast here, Natalie.
This is my pleasure. Thank you, Chris. So I usually feel painfully awkward making grandiose statements, so I will try to be personal and humble. And I would like to say that I'm extremely grateful and honored to be part of this operation of promoting and developing the domain of tensegrity in the larger field of research and science. Biotensegrity and tensegrity is a relatively new kid on the block. It is barely 6 decades old, which is nothing in comparison with the mechanics of Newton and biomechanics of Borelli. So, of course, biotensegrity as it is still developing its toolkit of experimentation and simulation, and we're still not fully fledged, but it is extremely rewarding when once in a while it helps to connect the dots and to reconcile the complexity and ingenuity and beauty of biology with the streamlined and elegant laws of physics and mechanics. So despite all these, well, I would say it's a fight to drive the concept to future students and engage people into understanding what tensegrity is, but it's extremely rewarding. It's like growing your own vegetables or parenting offspring. It's a fantastic experience, and I would like to say thank you to Steve, who was actually the founder of the field, was the first person with whom I had a very meaningful discussion as a PhD student, and he was extremely accommodating and patient and helped me to drive my own research in this field. So thank you.
Cheers.
Cheers.
Gambe.
Thank you, Natalie. And without further ado, I'm going to pass the baton over to Graham. Graham, will you please share our sponsors?
Yes, thank you, Chris. Well, you can find details of all our sponsors in the chat, but I'll summarise them first. Handspring Publishing in East Lothian, Scotland, have a wide range of authors and books available for movement and manual therapy professionals, and also produce a free newsletter which provides details of their webinar series with Elizabeth Larkham. Embodied Biotensegrity with Chris Marita Clancy and Gwen Cornish in Vancouver, British Columbia, who provide an excellent resource for those of you looking to learn more about biotensegrity. Join them next Wednesday for a Facebook Live event to hear their excited new news. That's a mouthful. The Second World Fascia Congress will be running a 3-day online event from Brazil on August the 12th to 15th. The fasciahub.com in the UK, who produce regular seminars on fascia-related topics. Integrated Biotensegrity with Lisa Babiuk in Alberta, Canada, who runs regular courses. Matrix Repatterning with George Roth, who also run regular courses. Artifact Pro in Madrid, Spain, who produce anatomically inspired tensegrity models. And in the model line, we also have Pretensed in Holland, who have a wide range of different tensegrities for sale. And Bruce Hamilton, who is a long-established tensegrity model maker. And you can find more details about all of these, our sponsors, in the chat. Thank you. Back to you, Chris.
Thank you, Graham. And it is my pleasure to be here with you, Natalie and Hila. One of the blessings that I get to receive as being a board member of the Biotensegrity Archive is to meet people like you. And as you know, the Tea Party is really bringing together people from diverse backgrounds and diverse fields. So Natalie, you are at McGill, you're an assistant professor in the Department of Bioengineering, and really at this edge of bringing biotensegrity into academic circles. And I hope we have a chance to talk more about that. I just want to let our audience know a little bit more about you. You've got a PhD in structural biology from the Weizmann Institute of Science in Israel, and has studied architecture of bone using 3D tomographic imaging methods that span size scales, ranging from atoms to the entire skeleton. And people here on this call tend to know a lot about biotensegrity already. And I know everybody is excited about seeing your research. And these scale levels are so important. Steve is always telling us when we get stuck on something to change scale levels. So without further ado, as you set up your PowerPoint and get ready to present, I invite our audience here on Zoom to shut their cameras off for now. That will give our YouTube audience a better visual. And also, if you're here on YouTube, hi, thanks for joining us. Please do let us know where you are from, who you are in the chat. Also, you can put your questions there, your comments there, and Neo is going to be transferring those over to my question and answer sheet. So at the end of the presentations, we will be able to share those questions with Hila and Natalie. As well, if you're here on Zoom, the questions can go right in the chat, and we have Patricia monitoring those, gathering them up. She's sending them to me, and I will forward them to Natalie and Hila when we get to question and answers. Okay, so whenever you are ready, Natalie.
Yeah, I think I'm ready, and I would like to start with a little preamble. And excuse me if I'm being too philosophical, but I just wanted to reflect on my own perspective, why biotensegrity is so precious for us as a research toolkit. So humans are big specialists in assigning meanings to things, whatever that would be. Sometimes we assign meanings and purpose to things that don't have purpose. Sometimes, well, that, for example, would be what for winds blow. There's no purpose. These are the way things are. But sometimes we find a superficial meaning that eventually evolves into more complex meaning and more underlying deeper purposes. So this is also a subject of your epoch. So for example, if you ask a medieval monk why flowers bloom, they would give one answer. And if you ask David Attenborough, he would give another answer. And it's a never-ending process in unraveling the meanings of things and eventually parsing the most relevant ones. So tensegrity equips us with a rich variety of tools of figuring out what is going on in biology and in evolution. So as a skeletal biologist, I will be talking about the meaning of bones, meaning of skeletons, if you will. And skeletons actually appeared in evolution about 500 million years ago, where it was a serendipity, it was a heuristic event, when turned out that by having a skeleton, an organism has better chances of finding a prey or escaping from being eaten by a larger organism. And then for terrestrial animals, it turned out that the basic building plan works the best. And this building plan includes, for example, having the head with the teeth and the sensors on the front end of the body, and the pelvis and the tail on the rear end. And because nature uses and reuses whatever works well and is good enough, we end up with the features that we share across all the clades of animals. And if we look at the skeleton of a mouse, we will find the skull with the teeth, we will find the forelimbs, and we will find the hind limbs and the spine. And this skeleton as an archetype, as an archetypal structure, as a blueprint for any other mammalian skeleton, fulfills many roles. It's a storage of ions, it's a protective shell for inner organs and the brain, but the main salient feature and the purpose of a skeleton would be to enable locomotion and propulsion against gravity. So I think we could agree, all of us, with the fact that this is the main purpose of the skeleton as a whole. Now, if we zoom into a particular element of a skeleton, for example, this shoulder bone of a squirrel, we will find something that we all share. Porcupines and elephants and humans and horses and squirrels. And this is the shape of a long bone, which is kind of a classic shape with the middle part being elongated and hollow, working as an effective beam in locomotion. The flank parts flare towards the sides where they articulate with other bones, and they're called metaphyses. And in all animals that possess endoskeletons, this is the basic structural plan. There is some peculiarity here in this squirrel bone that reflects its lifestyle, right? It looks like a screw, looks like a propeller blade with these ridges where muscles attach. And I find it quite stunning and even more interesting than my own skeleton, because I don't bounce off the tree branches. But in squirrels, we have this screw-like twist, the purpose of which apparently is to accommodate this resilient and failure-safe mode of moving around. Many bones actually feature this spiral twisted shape, like our own clavicles have the shape of a screw, and their ribs, they follow a twisting pattern around the body axis. So maybe there's also meaning in that, and having spiraling structures is effective for resilience and shock absorbing properties. But now if we look even closer at the inner structure of bone, and now for the sake of diversity, I will present another animal and another skeletal element. And this is the proximal femur or thigh bone of a sheep. So if we take a virtual cut and look at how the inner structures organized, we will find this very intricate architecture where tiny struts and plates all merge and split and interconnect into this enormously complex 3D lattice. And this lattice forms the bulk of this flaring end of a bone, which is located where bones connect to other skeletal elements. I will wind it back, and because I want to highlight again that in some parts of this bone, you can see very fine and uniform texture, and sometimes in some other locations, just a few millimeters off, you find these buttresses, these very robust and coarse elements that blend with the shaft that is hollow down this spiraling structure. So the question is, why nature evolved this intricate 3D scaffold or framework of interconnected beams? Or to paraphrase the question posed by Rik Huiskes, if bone is the answer, then what is the question? So my personal journey into the question of bone coincided with my acquaintance with biotensegrity. So back at the time at the Weizmann Institute as a student, I was looking at micro CT scans of human proximal femur, which looks quite similar to what we saw in the sheep. And as in the sheep, the interior is packed with this interconnected struts and plates. And I was wondering, is there any rule? Is there any mathematical underpinning why we have this spongy structure where the femoral head articulates with the socket, with the counterpart of the pelvis? And also we have trabecular architecture where muscles attach to the femur at the greater trochanter here and the minor trochanter here, and they apparently are being loaded in a totally different way. So, of course, we were not the pioneers of this research, and people have been looking at trabecular structure for centuries, and there are many parameters that are worth studying. So one can research the extent of porosity or extent of co-alignment or texture of these individual elements. But fresh and, as it turned out, useful concept that occurred to me was that what if we ignore the morphology and the shape of each of these interconnected elements and just look at the core of the matter? So imagine that in a fragment of bone, well, we have these many interconnected curved trabeculae. But what if we construct just one line, one axis, or the centroid of each of these elements and position it in space the way it is? Because trabecular network is all interconnected and continuous, where one trabecular split or merges with another one, we will have connections of these centroids. And if we continue plotting these central lines throughout the entire skeletal element, we will end up with this 3D graph or mesh that in a simplified way looks like a constellation. But imagine we have a mesh of 500,000 elements, and that is the number of interconnected branches in a typical proximal femur. So the question that we were trying to pose at the time was, is there a connectivity rule? Is there a mathematical principle unrelated to the thickness of these elements and to composition or the material properties, or in a more simplified way, is there a mean angle between any random pair of interconnected struts? The beauty of this question is that we can ignore individual variations that can be linked to the general skeletal constitution or body size or sex or lifestyle. And if we have a sample of very thick and massive trabecular struts and another sample of thin and gracile trabecular struts, if their connectivity rule holds, they may have the same blueprint. And perhaps the blueprint might be the key to this problem. If bone is the answer, what is the question? In some sense, this topology or blueprint or Bauplan defines the way things operate. And to make a more relatable example, let me present, a pair of trousers. What about their topology? Well, a pair of pants has 3 openings, one for the torso and one for each leg. Regardless of the material and style, all trousers have the same topological plan. If we close one of these openings, or we will add an inappropriate 4th opening, pants lose their functionality. And that it doesn't matter what is the material and what is the fashion style, they don't fulfill the job of being a pair of pants. So maybe we can extend this logic and look at the topology of bone and figure out what is it for. So how do you build the topology of bone, or the graph, or a skeleton of the skeleton? So as I said, we can construct a centroid in each and every 1 millimeter long, 200 microns thick strut. And at every position where 2 or more trabeculae connect, we will place a node. Then, once we disregard the shape and size of individual elements, we will have this graph, a 3D mesh where every edge stands for the position in space of our individual trabecular element, and the node is the connection of several elements. So once we have this structure, it actually makes it mathematically feasible to figure out, first, how many neighbors every node is connected to. Are my nodes connected to 3 nearest neighbors, such as all the magenta circles in this drawing, or maybe they're connected to 4 nearest neighbors as the teal nodes? As well, when we consider, let's say, any pair of adjacent trabeculae, we can construct an imaginary plane that goes through their shared origin and their individual separate termini. In that plane, we can apply formula and calculate the angle. And this angle, if we repeat this calculation statistically over the entire, let's say, proximal femur, we may come up with the general meaning of what is it for. But before I dive into the complexity of real bone structure, let me give a little bit of a theory about what we can expect from very regular lattices. What are we measuring in general? So, for example, if we look at a very simple 2-dimensional symmetrical honeycomb lattice that has hexagonal symmetry, if we assume that this lattice is infinite, then all the nodes would have just 3 connected neighbors. and the coordination number for any random node will be 3. If our honeycomb is symmetrical, then all the angles in this infinite array will be 120 degrees. And these two descriptors would be sufficient to reconstruct this honeycomb at any scale and any size. And then we can dress these individual edges in any thickness, and we can render them in any material. But these topological parameters would remain the same. But it's fairly easy and intuitive in 2D, but what if we switch to 3D? And the simplest symmetrical and space-filling structure would be a 3D diamond lattice. In a 3D diamond lattice, all the nodes have 4 connected neighbors. So every node in an infinite array that follows this diamond symmetry, every node will have a coordination number of 4. If it is a symmetrical structure, every angle in the array will be exactly 109.5 degrees, which is the classic tetrahedral angle. Now in 3D, of course, we can connect nodes in many different ways. And maybe more familiar to us, because this is the way we build houses and furniture, is the 3D cubic lattice. In an infinite array, such as a 3D cubic lattice, in the simplest form, every node will have 6 connected neighbors, and all the angles would be 90. We can, of course, connect these nodes with a larger number of edges. We can connect them diagonally through the cell or across the diagonal of every cubic face. But that will increase the complexity and the connectedness of such a lattice. Now, it turns out that in bone, you can find examples of all these symmetrical motifs. You can find nodes with 3 connected neighbors. You can find the nodes of 4. You can find pentuple nodes, and sometimes you can find nodes of 6 interconnected neighbors. And here, these are cropped examples from a micro CT, computed tomography image of the same sample of bone. So it's a portfolio of a bone lattice. Now, the interesting thing is that for every type of nodes, the angles that are established between adjacent neighbors are not exactly strictly symmetrical. They rather form a distribution which is fairly broad. But for a triple node, for all the population of triple nodes in a bone lattice, the distribution will center around 120, will be very close to this value, which tells us that these elements tend to distantialize from each other as far as they can. Now, for quadruple nodes, this distribution, which is quite hairy and broad, but it's predictably creeping to the left. And now for quadruple nodes, the center of the distribution will be very near 109.5 degrees. For nodes of 5, the average offset between emanating edges would be around 100. And it's important to note that mathematically it's impossible to offset 5 edges at the same angle with respect to each other. But on average, that would be around 100 degrees indeed. And in the case of hexaple nodes, we will have the center of the distribution around 90. Well, another I find it a fascinating thing is that in a bone lattice, we have different proportions of the nodes of different complexity. So the champion in this lattice, the main structure forming element, is the node of 3, is the Mercedes-Benz star, where 3 nodes are offset at approximately 120 degrees. These nodes form more than 3/4 of all the lattice-forming elements. Less than a quarter are the nodes of 4, the tetrahedral motifs. Very rare are nodes of 5 emanating edges, and nearly impossible to find are the nodes of 6. So if we want to describe this, this ratio of nodes of different coordination number, we can form something that looks like an exponential decay graph. The more complex the node, the more difficult it is to find it in the bone lattice. So we can see from this graph that the lattice of trabecular bone is way less interconnected than even the diamond lattice, and even more complex than the cubic lattice. And therefore, we can call it the sparsely connected lattice. And obviously, the maximal offset holds for every type of nodes. We were very excited about it. And we wrote a paper that presented this parameter in several samples of human proximal femur, and we looked at the femoral head, at the trochanters, at the neck, and we found the basic topological blueprint. And then I took it further and I looked at some museum specimens of human bones that I found in London, in the UK, and these specimens were from ancient China, where a common practice was food binding to restrain the growth of female feet as a standard of beauty. So that was quite an incapacitating practice, and the individuals affected by this deformity couldn't really walk far or fast. They had all the entire package of bone tissue deficiencies, but the topological blueprint was the same. And then with the Weizmann people, we compared this trabecular blueprint that we found and described in humans with the same motif in the pig bone, in the mandible, and in the femur of the pig, and again we found the same. So things started looking suspicious, and then Grun and Nebelzig conducted a similar study on sea urchins. And sea urchins is a different clade of animals. These are echinoderms. Their skeletons are not even built of calcium phosphate as our skeletons. They are made of calcium carbonate. And their skeletons that are quite porous and trabecular, they follow the same rule. These networks are sparsely connected, and the main space-filling motif is a triple node with an average offset of emanating edges at 120 degrees. So why? What is the story? Why things happen the way they are? What is the question to which trabecular bone is the answer? And I think we managed to dig out quite a feasible explanation fairly recently, where this young scientist, Amar Al-Shegri, was doing his PhD at McGill, and we collaborated on the topology of bone. And we decided to go for a simulation study. So we knew the descriptors of natural lattices that are all the same, regardless of the species and anatomical location and the mode of loading. We know that they are the same. And then we decided to build a proto-lattice, or the mother of all lattices, the lattice that has way more edges connecting neighboring nodes. And we subjected this lattice to an evolutionary algorithm. So like in evolution, an organism produces numerous offspring that have some minor genetic heterogeneity. And those that are more fit for the environment, they survive and propagate their minor variations in genetic pool, and the rest is being eliminated. So the same thing we did to the mother of all lattices. And the lattice was subjected to simulated mechanical loading, and the connections that were redundant or idle or didn't bear any stress, they were thrown away. Our idea was that by doing so, we will establish a pedigree of lattices from the superconnected proto-lattice to lighter and lighter and lighter simpler lattices, because we know that in natural lattices, the connectivity is very sparse. So the lattices of different generations were compared against some preset criteria. And we were just wondering at what stage, at what criterion we will reach topological similarity with the lattices that we can find in natural bones. So the way it worked was the following. How do you construct the mother of lattices? Well, you take the natural lattice from a scan of bones, and those I had plenty. So we make a lattice that is made of nodes and connections. And then you discard all the connections and only keep the nodes, because the nodes are most important. Now, if you reconnect the nodes with a kind of a more generous allowance to reconnect, let's say instead of connecting only to 3 or 4 nearest neighbors, you can set the criterion, please connect to 8 nearest neighbors. So then this lattice would be denser than its natural analog. So this overconnected structure would be the proto-lattice that will enter this cyclic process of evolutionary optimization. We loaded it in simulation, spotted the elements that were idle, that were not bearing any load, and disconnected them. And then subjected it to another cycle of testing, figured out what other elements were still idle, and threw them away. And along this path, we were checking what is the minimal bending stress experienced by the lattice, when can we get to the equivalent weight or the volume of elements to which you can assign some nominal thickness, and when can we get to the equal number of connections per unit of volume, as in the natural structure. So this is the pedigree. This is the mother of all lattices. Oh, sorry, this one, the naive, is the proto-lattice. And these are the generations that answer the exact criteria that we posed at the beginning of the study. So in this one, generation 1, the number of elements was like in the reference lattice derived from bone, but they were connected in a slightly different fashion. In the generation 2, we checked for stiffness, and for generation 3, generation 3 had the equivalent mass, and then for all of them we measured the prevalence of stretch and bending strains per element. So to compare, because topological descriptors are kind of bulky, and it's difficult to describe it with a single criterion, we came up with a parameter that you can calculate from the graph, from the abundance of nodes of different coordination number. So in the reference lattice of bone, the decay coefficient, the parameter that tells you how much this graph sags between the highest point and the lowest point. That decay coefficient, a single number, would be minus 1.5. In the overconnected naive lattice, before it enters the cycles of evolutionary optimization, the nodes of high valence would be more abundant, and therefore the graph is a straight line. There is less of simple elements and more of complex elements, so the decay is 0 because it's a linear graph. Now, as we go ahead and remove element after element after element in the cycles of loading, the abundance of simple nodes starts going up at the expense of the abundance of more complex nodes. So the graph starts sagging. Its decay coefficient becomes more pronounced. So here the decay is 0.7 negative. As we proceed to generation 2, we have even more of simple triple nodes and even less of complex nodes. And now the decay coefficient is -1. And then when we arrive to generation 3, we have quite a similar proportions of simple nodes, triple, quadruple, quintuple, and hextuple, as it occurs in the reference lattice. So this is a kind of a maybe overloaded graph, but I will walk you through. So this is a simple intuitive process that as you throw away elements, you start with a super connected lattice, then you go down by throwing elements out, and this is your generation 1, and then generation 2, and generation 3. And the reference lattices derived from natural bone, they are very near generation 3, right? What is also quite surprising to me in this graph, that if you compare different natural lattices, they all cluster around the same complexity that is expressed in this decay coefficient. They all form vertically, they all line up around the same value, although the number of elements could be different. But this is what was going on during evolutionary optimization. Now, if you ask this question, what if you want to optimize the structure for maximal stiffness at the minimal metabolic expense? What is the stiffest but the lightest lattice? Where would that land on the graph? And here we have plotted the stiffness against this progression of optimization of lattices. It turns out that first generation has the highest stiffness normalized by weight. So this is the answer that would be, should the question be maximal stiffness at the lowest weight? But our evolutionary progression went down the slippery slope and actually converged with the natural structure, with the reference lattice of bone somewhere in the bottom right corner. So apparently there is no limit for stiffness, and stiffness is not the target criterion in the evolution of bone architecture. And then the answer was actually in the graph of that describes the combination of bending and stretch-dominated stresses. So let me explain it as well. So we start with the overconnected proto-lattice, where this low value tells that most of the elements in the lattice are loaded in compression or tension, which is a wonderful property, because if you want to break something in compression or tension, it will be fairly difficult. It's way more easy to break things in bending. If you try it on a pen or a pencil, you will see for yourself. So our overconnected lattice had a very pronounced domination of compression and tension at the expense of bending. Bending was not allowed in this lattice. as we keep throwing away idle elements and arrive to the generation 1 lattice, it is still dominated by tension and compression, and bending is not allowed in this structure. When they progress to the generation 2, this lattice has even less elements, and the nodes of low connectivity dominate or start dominating. We still have a relatively little occurrence of bending stresses, so this lattice is still dominated by axial strains, which are beneficial. As we progress further, generation 3 lattice and generation and the reference lattice derived from bone, they're still on this shallow slope that is indicative of a very biomechanically favorable stretch-dominated behavior. And if we extend our evolutionary optimization by throwing more and more and more elements, all of a sudden, bending stresses skyrocket. So it looked like a maybe not a coincidence that the natural lattice of bone sits exactly at this corner, at this inflection point, where we have this general behavior of the architecture as stretch dominated, where most of the stresses are compression and tension, but not so much as bending and torsion. But if you overcome that boundary, all of a sudden, loading becomes dangerous. So from that, we may conclude that trabecular bone is the lightest structure that is not the stiffest. It is still stretch dominated. And stretch domination is independent on the absolute mass of bone. And apparently, this design evolved in articulating ends of bones near joints to facilitate stable locomotion and fail-safe mechanical behavior. So to put it in a lay term, bones are cheap and reliable. And that's, as I understand of today, that's the answer, the question to which bone is the answer. So that's all for now, and I'm happy to take any questions.
So thank you, Natalie. So interesting. And we're going to actually take the second presentation from Hila first, and then we'll do questions at the end. But fascinating. And I see those images and they're repeated over and over again. And we also see these same images when we play with bubbles and so on and so forth in the fascia. So I know we're going to have a lot of questions. They're already here. They're already piling up. But I would like to move on and introduce Hila for their presentation. So Hila Tzipora Chase is a multicultural artist and scientist currently living in the traditional territories of the Salish, Pandorial, Kootenai, and the Blackfeet people in the heart of the Rocky Mountains in Montana. They're currently finishing their PhD in evolutionary biomechanics and ecology of bird flight at the University of Montana. Hila's work focuses on roots-based integration to provide hands-on approaches to systemic reform, decolonization, community building, and eco-psychological health. They have always based their work in fostering deep integration, embodiment, and interconnection for all. Now, before we move on to your presentation, Hila, I just want to let everybody know that Natalie and Hila have worked together on some of their projects. That first paper, I believe it was, Natalie, that you put up was also, Hila was also working on that paper.
Yeah.
So
I'll
do you want to
be
take it away?
mentioning that.
Pardon?
I'll be mentioning that, don't worry.
Okay, awesome. Well, we'll turn the baton over to you, Hila.
Awesome. Okay, let's just make sure I can share this properly. So welcome and thank you, everyone. Tell me if you're seeing this. Okay, so I'm really excited to give this talk. I'm really excited to give this in conjunction with Natalie's talk. And as Chris just said, yeah, Natalie is actually the reason that a lot of my current research is based in trabecular bone and is a huge part of how I got into biotensegrity in the first place many years ago. So as a very, I think probably 7 years or so throwback, when I first came to Weizmann, I was an undergraduate doing a research internship, and Natalie was assigned as my mentor. And so we started talking about bone structure, and we got into developing this intertrabecular angle approach. We had a lot of fun hands-on things, not just very embodied from the beginning, I would say. So not just like coding and screen-based and visual analysis-based things, but really just like thinking through, talking together, and it was really, really incredible and formed a lot of the basis of my research background. And we have kept in touch and like to have fun together as well. So really excited to kind of continue all of this together. And so I'm gonna have like a few throwback slides. This is literally slides from the presentations that I did on that research project way back when I was still an undergrad. And so much of it was really beginning theoretically in this biotensegrity approach. And so we were looking at these angles and thinking of bone and thinking at various different hierarchical levels of the materials and why these kinds of models made sense for what we were seeing and measuring in human trabecular bone. But then we also, you know, at this early stage, we're really a lot of the discussion was about where trabecular bone was at in biological research, a lot of which was Wolff's law or mechanical adaptation. So the overall observation that this complex matrix in bone will shift its three-dimensional structure in relation to the main directions that force is applied to bones in nature in an organism. And so when we were looking at this biotensegrity structure approach, we came up with some fun metaphors of like, even though this is this three-dimensional tensegrity structure, you're still seeing thickening perhaps. You're still imagining it like cross linkages where you have tree trunks in a forest and branches between. And the important part is that the branches also contribute to the whole structure's function. So as if the trees were actually interconnected, which in a different sense they are, but not mechanically. And a lot of this was coming from research that I was already doing in paleontology and comparative biomechanics, in this realm of looking for patterns and looking for signals, but not actually questioning very much why they were that way, how they functioned, what the mechanics were. And I was already really into Buckminster Fuller and a lot of sort of systems theory and, you know, paradigm shifts that see reality as a network, not as categorical and as interconnected and dynamic. So it meshed really well with all of my interests and kind of set the stage. But what also set the stage was just sort of my family and general background. So when people think of art and science, it's always interesting to me what they think those terms actually mean. Because if you're totally deconstructing like our categorical fields and disciplines and systems, then to me, I was raised with art and science just kind of being part of this cycle of how things work and how to engage with the world and figure things out and create, figure more things out and just keep going. So rather than seeing art and science as two distinct fields, mostly I think of art and science integration as really just another way to describe integration across the entirety of reality or within a totally systemic paradigm or network-based paradigm. So when I was an undergrad, I ended up developing this sort of roots-based integration paradigm. So over time, this is coming from the research I did at Weizmann, from so much else that I did throughout my career, and kind of culminated in this model or paradigm, which itself is a model and paradigm against using models and paradigms. So we do what we can with our human language. But the idea is that if you consider these trees to be two different disciplines or two different things, really anything you're trying to think, how do these connect? How could I integrate them? Or how are they integrated? The model is simply saying that instead of building artificial bridges between artificial categories, if you really investigate things, they already have roots. They already have ways that everything already has a way that it connects naturally. And so if we force ourselves to go deeper and be detail-oriented and attentive and look at where the roots are actually intersecting, those are the things that we can use to do integrative design, to investigate things that have multiple different like fields of research or media required. So we can actually guide ourselves directly based on what exists instead of trying to force it into categories. And so the basic thing that I tell my students is overall rule is just find where the roots touch between anything you're considering, be it within a specific scientific project, or if you're integrating like art and science. There are a whole lot of different interdisciplinary design courses that we teach and workshops, and it ends up being really helpful for students to just have this really simple rule to start with. And so I want to also make sure to kind of push the point that in a relational or integrative paradigm, a really important part of understanding how that actually operates is that it is practice-based and participatory and embodied. So we can conceive of concepts and paradigms and they can be abstract, but actually using them, actually having them be applied to reality means that we are a part of them and that we're considering everything like we are organisms considering other organisms and material. And so when we think of knowledge this way, we want to kind of think of knowing or knowledge as practice-based. So we go through acts of knowing, of observation, of learning, and then we put them into knowledge systems, our models, our paradigms, things like this. But that always should hopefully go back into how we are still updating that and not getting trapped in knowledge systems or paradigms that don't actually update toward reality or consider things in actuality. And so a lot of the time I apply these things easily in outreach and education and have a fun time. I love working with kids and adults and various communities. And so much of this is embodiment and practice-based and hands-on. And going even deeper, we'll teach evolution through embodiment and through the very, very literal, actual connections that our bodies have to all other vertebrates, to other extents, even invertebrates and beyond, and to really, do exercises that actually teach this kind of thinking, but not so much in the head thinking, but in the body thinking. So really showing this, it's a great way to do your vertebrate anatomy labs or any kind of thing that we usually see difficult for students because it's full of memorization and there's not a lot of context to the structures and bones or other things that you're trying to know comprehensively to understand a system. So it helps a lot. And a lot of this work has gone into an overall approach of this roots-based integration paradigm. So there's the overall theoretical paradigm, and then there's a course I recently taught for a graduate seminar of like super hands-on approaches to doing this in your own work. We had probably 9 different disciplines at our university represented by a nice small cohort of graduate students. And so we went through kind of all of these things theoretically, but the real point of it was to have hands-on design practice, teaching practice, and research practice. So as much as we can talk about things academically or in settings like this and discuss it, a huge focus of mine is how do you actually train and provide sort of instructions for people to really practice this in their current jobs and systems and research projects and creative projects that generally are still very limited because they're not always as open to a super integrative paradigm like biotensegrity or any other holistic one. And so a large part of this is also equity work at a human level where all of this is effectively already pretty standard in Indigenous knowledge systems and traditional knowledge systems. And so when we have this big movement of diversifying the sciences, diversifying our systems, redesigning our systems to be more equitable, we're thinking at this really deep level of how people see the world and how people research and understand reality. And so much of everything I just talked about is in like thousands of years old. It's not new concepts, it's not new theories. It's just applied within the Western system, usually academically now. But this is a lot of the decolonization work that we're trying to do within the sciences and kind of across the board, where it's not so much that people happen to discover things that now science actually vets and proves. It's that there are complex, nuanced paradigms and attitudes towards reality and structures to see how things work that have been around for a very long time. They're just not very privileged in the dominant system of academia. And so when we talk in science about necessary paradigm shifts and integrate this in, we are really trying to get at the specifics. So whatever field you're in, whatever big think piece you're going to write, it's important to bring it all the way back to the details and the actual factors of reality. So in basic research, how you're actually setting up your experiments and your tests and your measurement tools and the statistics you'll use to analyze all of that, trying to bring it to the very basic level. So from abstract to actual is the idea. And so I try to do this in my own PhD research, like the side of just basic, straightforward research. But it's part of how I do outreach and education and reform work and kind of across the board work. So I don't really like to define things in categories very much, but it's important to be able to do these things in absolute deep detail. So if I'm teaching the concepts behind my research dissertation, for instance, when we think of evolutionary biomechanics, and I know a lot of us are probably used to thinking of biomechanics in a human way. So there's a whole lot of models, there's a whole lot of things in health, in all of the health sciences, and also all of engineering, which is also very human-based, because we're thinking of what humans need for our systems to function, whether it's building a bridge or a prosthetic. But we have a whole lot of other creatures that we share the planet with, and all of us are under the exact same rules and processes through evolution, through physics, that everyone else also has going on. So if we want to understand more about our own bodies, it's not just earlier the homology-based, evolutionary-based embodiment to teach these concepts. It's also important to understand how evolution actually works, how vertebrates actually evolved, especially under this sort of question of, or answer rather, of if bone is the answer, what is the question? That's a really interesting thought exercise to consider comparative biomechanics and evolutionary biomechanics, not just thinking of human bodies. And so when we look at how our own ancestors came out of water and evolved a skeleton, and eventually even could fly, you know, we kind of went on this other trajectory where we're really good at walking and running and we're weird and lanky, but there are all these incredible ways that skeletons have evolved. And so if we focus in on the changes in joint morphology, changes in kinematics or movement, and sort of wrap that all up into thinking about what we're actually seeing in evolution, whether it's ourselves or other species, we can get a little closer to applying this very integrative paradigm to the typical ways that people study these questions in evolution or paleontology, or even like bio-inspired human biomechanics. And so to do all of this traditionally in research, we always think of behavior of an animal or a human, whether you're in manual therapy or looking at bat flight, to function. So the specifics, the kinematics, the movements, the more specific level of thinking of a behavior. And then we have to think of a biomechanical model for that to make sense in. So very simple ones. We have moments of area, or if you imagine something bending, you have force applied downward on a beam. So you get tension at the top and you get compression at the bottom because it's bending. And you get this line through the middle that basically defines the moment of area. So as a very simple example, this works for I-beams. That's why we build with them all the time. But it also works for tubes. And in the vertebrate world, in the bone world, when we think of bones, a lot of the time we think of long sort of tubes filled with marrow. And a lot of research for many, many decades, if not centuries technically, has been all under the sort of model and framework of just thinking of second moment of area, just thinking of how the very middle section of a long bone will have thinness and diameter and these kinds of geometric components that should have something to do with how well the bone functions, how strong it is, how it's being used. And that's really helpful, and that's gotten us pretty far. And we see some really easy examples for my research. We often start with talking about how lightweight bird skeletons are. They're not actually lightweight relative to their body weight, but what they are is a lot thinner in wall specifically. So the walls of their bones are thinner. They still have pretty big bones, and they still have a lot of bone material in their skeletons. Now this is one of those like pretty simple models, sorry. Then we have biotensegrity, or just tensegrity in general. And this is interesting because it is, in human biomechanics, applied to limb systems a lot and applied to the spine. And I haven't actually thought about it that way so much in my research because we've been thinking of these internal matrices of bone and how that works as just a cellular solid. But when you take that into the overall body plan and you're looking at tensegrity, it's a very different kind of consideration. And it's not super popular in comparative biomechanics yet, but it's actually closer to being used more regularly in animal studies and comparative biomechanics than it is in medical research for human biomechanics. So it's an exciting paradigm for sure to apply across the board. And then of course, as I said, there's cellular solids, which is a little bit more straightforward as a model because we're just thinking of these chunks of bone, let's say, inside of bone and how that's working. But we want to go beyond these different models, and we also want to go beyond using them instead of each other. So there's a way to integrate all of these models and many more in a holistic biomechanical system. But to actually do that, we have to figure out what to test, what to measure, and how to contextualize those results with whatever model system or experiment that we're doing. And a big risk in this is that in science, we often get pressured to use things that are kind of simple models because it's already complicated and we're just trying to answer one question at a time. And a lot of the time this leads to assumptions that are not really detail oriented. So you're taking a basic approach to how bridges work or how engineering concepts work, and then seeing a biological structure and saying, okay, that looks close enough. Let's see if it works like that. And that's fair to start with, but it's only a model and an example. And what often happens is that between let's see if it works like that from a scientist and let's actually test and see if it works like that, we'll go into methods and approaches that are a lot more not really detail oriented and not really biological. So if you see the structure and it's close enough, and then you 3D print something that is just modeled the same way that, you know, any tube, like this is an example from bird bones where people are talking about soaring versus flapping and airplane design and these basics of mechanics. And you do observe that there are these really insane differences in bird bones, but they just took two bird bones, or two species of birds, one bone to focus on these kind of simple structures in the first place. But if you really look at them, there's a lot of complexity there. It's a lot of morphology, a lot of form, a lot of structure overall that's not being described when you go and simply 3D print a tube with struts that are fully uniform at various angles, and you just test what effectively has already been answered by your model that you're using. So this is not really pushing forward our knowledge so much as confirming what in some ways is a bit of a simplistic bias. And so to actually test whether these principles are occurring in bone and throughout evolution, to learn from it beyond the engineering that we have or beyond the understanding that we have of how things work, we need to go a lot deeper. And it's not just going deeper, but we need to always come back to the biological context if we're going to use these fancy scientific methods. So really recently, my lab came across this study that was published just this past year, and it's exciting because it's in pterosaurs, so giant flying, not dinosaurs, but pterosaurs. They're very related. And all these questions about how they could fly, some of them were as big as planes, all this skeletal evolution craziness, like how could their bones support these things? They're so thin, they're so giant, but they could fly. It really puts you at this intersection of pushing bone design or vertebrate evolution to its limits. And so in this study, it was really exciting where they did beautiful, full, detailed CT scans of a vertebra, a neck bone in a pterosaur. And they saw all these struts of trabecular bone and didn't actually detail how many trabecular struts there were in the actual sample. They went and did a bunch of computational work in this simplified model, so you can see these are just lines. They're no longer the morphology of the bone. They're not technically in the same position or architecture, but it's close enough. And then they tested all of this computationally and came up with some really cool results about the number that contributes to the vertebra overall being a lot stronger, but also potential trade-off where the neural tube in the middle, very thin bone, had increased tension and could fail if there were too many struts. And that's a really cool concept, but it was a bit disappointing when we went through the paper to then get to this point of having a really good framework, a model, detail-oriented analysis. They have the data, but the next step would be plugging it back in and actually looking at the genuine trabecular architecture of these and a little bit more detail to understand what is happening before you go and make a big evolutionary claim or say that it's an adaptation for this or that, because there's so much detail that you're still missing. So in my research, I look at birds a lot. They're awesome, but also because specifically of all kinds of animals that we still have alive today, birds are like really pushed to the limit when it comes to skeletal evolution. And specifically, we know that skeletal evolution and bones, bone biology and changes over time is what allowed dinosaurs to evolve to become birds and become able to fly and do all these things that are pretty difficult and kind of wild from a mechanics and engineering standpoint. And an experiential standpoint. We're pretty good at like walking and running, but flight is always been a huge inspiration for me and sort of how I contextualize the way that I like to move and embody things. And so in general, what we basically have here is a really good model system for deep evolutionary questions, deep questions about bone and why bone is so highly conserved across so many different animals, and why it seems like bone is responsible for the largest animals that we've ever had on this planet, and some of the most incredible ways of locomoting, and just all of these examples of amazing biomechanics that do seem to be facilitated by the evolution of bone as a tissue, as a material, and as a system. And so when we look at all of this in birds, there's a whole lot of assumptions made in birds. I think it's very popular to think of lightweight bird bones. There's a lot of misnomers and misunderstandings. When you actually look in, we have to set up a paradigm or a system to test some questions against. So in my research, I look at how flight evolved and apply the substructure, the trabecular bone and the shoulder morphology to how it actually evolved to be that way. And so at a very simple level, you can look at the difference between holding your arms out, let's say, or flapping. And that's a very different range of motion, very basic level. But if you get into the details, you can start looking at the ligaments, every attachment, every piece of bone morphology, every detail, and you can start to model what you think might be happening. But the important point here that has formed a lot of how I think about how to research and this hands-on approach is that when I was first doing this, setting up my dissertation, I didn't really have a lot of research to go on. I was just sort of modeling from anatomy. And at the end of the day, the methods that I chose for measuring, or the sort of classic subfield that I'm in, you can model this all you want, but if you're only going to be measuring sort of architectural, highly detailed things about trabecular bone, then you don't necessarily have the ability to connect what you're measuring to explanations here. This is sort of the best I could do at the time, but a lot of what I've developed since is a lot more holistic. So in a holistic approach, seeing bone as a continuum, the point is not to measure a chunk of a substructure and then measure the midsection, like the cross-sectional geometry, and then measure how the bone functions in the shoulder, in the body, as separate things, but to instead try to consider all of them at the same time and figure out the right kinds of measurements to make that will lead to answering these questions within the context of this integrative paradigm. And so, like, when you look at the bone, cortical bone, for instance, you're seeing that there's still all this matrix inside and they're not separate at all. It's a continuous tissue. More to the point, because I guess I like including animations. We're gonna just skip ahead over here. But when this, as an example, when we do these measurements, what I'm really talking about is very specific, highly detailed measurements of the thickness of trabecular bone, which is how thick each element is across the sample, or how rod-like or plate-like they are, so it's kind of a structural description. You can look at the anisotropy or the directionality of each element, and you can look at the volume fraction, or sort of basic cellular solid measurement. But all of these parameters, all of these factors that you can measure are only as good as you can understand why they might be that way. And there's a really big tradition in trabecular bone with comparative work of not really thinking about why they're that way, just trying to find patterns. So if you have like jumpers and runners, you're talking squirrels or any other kind of animal, as long as you can statistically show that there's a difference in enough samples, that's a paper, that's an answer, but it's not getting at why things are that way, of how they function. So throughout the dissertation, I ended up developing a new parameter for trabecular extent, and a big part of this in birds, not mammals so much, but in birds became necessary to separate the matrix from reinforcing structures, or struts. And so these appear very differently in a lot of birds. In mammals, it's a little bit more of a continuous matrix. But what was really interesting throughout all of this is that we found a lot of patterns based on that measurement. And we did the exact same measurements using a very typical classic cross-sectional geometry approach and didn't actually find any functional patterns. We found a little bit of patterns that have to do with aquatic adaptation, and that's a whole nother cool story about bird bone. But what we learned so far from this is that not only is this a good parameter, this trabecular extent, or way of like understanding how much trabecular bone just in the first place there is within a given long bone and the character of it, but we would have totally expected from any study perspective to find evidence and patterns in cross-sectional geometry, and we didn't. And so it's this really interesting example of doing something harder that usually produces worse results, and then trying the tried and true easier method in science, and usually you would expect better results. At the very least, that's what you might use going forward, but this time it didn't work. And so we kind of put this into the perspective of actually looking at the variation. So this is maybe a third of the different bird species that I have scanned, so very highly detailed micro CT scans, but they're super different across the board, not just in the tube structure and the thickness of the bone wall and where these sort of substructures appear and the character of them. They're just extremely varied variable. And so if you're going to actually measure the variability of this and try to look for patterns and understand why they're like this in a functional context, you have to really directly measure what is happening to them and be able to test that, and then again put it back into a natural context. And so instead of doing models, one example, a sort of small project I'm running, is taking specific sections of the reinforcing structures and putting them into a classic materials testing format, 3D printing them, and looking at these results in a comparative and evolutionary framework, not just an engineering framework. So we're not just asking, like, if something fails faster or slower or better or worse. We're looking at a specific context of how each of these birds flies and where in the bone the section is and how force is probably distributed in that section. And then statistically putting that into a phylogenetic context where we have like 50 species. We're probably not using all 50, but like even with 10, being able to compare these things allows us to get more detail about how to actually answer the how part of the question, not just the what. And it does it in a way that's not already assuming the answer in the way that you're asking the question, which is what we get caught up in the just so stories and the mechanical models that are too simple. And so kind of just a fun look at how that's going right now. But this is like slowed down a lot already, but we have an old materials tester and we're just kind of chewing through bones. And this was a lot weirder than I thought it would be because the computer is from the 90s and I had to bring the hard drive all the way to New York from Montana just to read the data off at what time. But this is the kind of thing that leads into what I mentioned before, which is a fully holistic approach to understanding bone as a continuum. So in biotensegrity, I think we're used to thinking about bones and ligaments and muscles and tendons and the fascia and how everything integrates as a system and as a body. And so in the same sense, to me, when you go deeper and you look at the details of bones, it's the same kind of approach, but you're looking at just a bone, let's say. And so when you're considering the geometry and the trabecular matrix and any kind of material properties or cellular processes, you're considering it as a continuous live tissue that is also attaching to those ligaments and muscles and tendons and all of these processes. And so whenever you measure any part of this, you should have, I like to say that you should have to force yourself to put what you're measuring and what you're finding out directly into the context of everything else and make it make sense. And if you can't make it make sense, that's exactly where you might need more investigation or more description, more testing, or you might need to revise your understanding of how things are working. And so to be very, to try to go really deep and roots-based right now, what I was excited about so much from Natalie's more recent work with this algorithm and the talk she just gave beautifully is this really nice example of roots-based integration, of what I mean by that, of, you could say, roots-based versus bridges-based. So when we're thinking of networks and nodes, we're thinking of this actual trabecular matrix, right? But we started out talking about systems theory and relational and integrative paradigms. And so thinking of reality as a network where everything's interconnected and you don't have categories, you just have interconnections between things. And so when I give talks, and when I first started giving talks about like relational theory or systems theory, or a lot of the things that feed into biotensegrity in general, I always pop up a network graphic. And a big part of what I do is try to get people to key into the idea that when you have a network represented like this, the stuff, like the actual stuff of reality, is the connections, not the nodes. So there we go. So if, the nodes are just where things happen to intersect or connect, but the connections are more abstract than the nodes are more abstract than the connections, sorry. So that means that like these things you're seeing now, the nodes are not as real or actual or direct and physical as the connections. And so because if we're looking at reality in this paradigm as dynamic and practice-based and all of that, it means that nodes might shift and change. But that's just describing how things relate to each other and work. And it's a bit of a deep concept thing, but tends to go a little bit better. I'm just a little tired today, but what I found really exciting about this is that in discussing the topology of trabecular bone and in thinking of the algorithms that are developed, I found it really interesting to think that step one is to look at the nodes and that basic topology, and then using those nodes, iterating through the algorithm as Natalie described. But in reality, the nodes are something that is changing, and it's secondary to the connections, the actual stuff. And so if you think of the trabecular matrix, if you think of the development, so this is throughout ages of how that matrix develops, then you actually see, because it's a continuous material, that yes, the general topology will probably be similar over time, but if we're thinking of evolutionary time, we're thinking of development, then really it's the connections, the struts, the trabecular elements that are changing and growing and remodeling and forming different nodes over time. And so it's not just a metaphor or an analogy here, but on one hand, we have a paradigm of like an entire perspective on reality and how it works. On the other, we have a really specific example from nature of how our bones work and how they're structured and what's actually going on inside. And in both cases, we're looking at nodes being secondary and the connections being primary. And I think this is really important because functionally, when you consider how to apply things, the important part isn't what it is, but it's how. It's how it works and how to test against that. And so if I'm designing a project or research or a show or an art piece or anything, and I'm thinking root space and I'm thinking, how do things already connect? I'm looking for those nodes, but I'm recognizing that the content, the actual things, are maybe easier to measure without understanding where exactly they connect yet. And being able to say, okay, I know that node exists, but there might be all these nodes that we're not sure of. So you can model nodes, but what you can't model is actual material reality, the connections. So sort of parsing that apart, I think helps a lot with really deeply integrative design and approaches to research. So that is all that I have for you today, and I would be excited if anyone wanted to check out further reading. I always pop this pile of books up, and I'm also really happy for folks to reach out if you're interested in like workshops or collaborations, but especially lately I've been trying to do this design approach a lot more simple than the concepts I went into today for various groups for pedagogy or equity training or things like that. And yeah.
Thank you, Hila. Very exciting to see your work and a toast. We're gonna have Q&A before we go to the toast, okay? Very exciting to see your work. And actually next week we have Neil Galloway talking about evolution. So it's a perfect segue into that for people who are also here next week. I have so much I could say. And here's Susan. I think she probably wants to say something. We'll invite people to turn on their cameras now. Susan, do you want to say anything before I keep going?
No, thank you. I mean, it's just that thank you guys. So exciting, so wonderful. What I'm hearing from both of you just fires so much and so delightful. Thank you.
And also this approach of the roots-based integration, I believe will really appeal to a lot of our body workers, our movement people. And then to have Natalie here to bring the perspective of the academic side. We've got a lot of questions coming. Natalie, I know I want to be really conscious of your time. And you presented first. So I'm going to begin with questions for Natalie. Okay. And the first one is here. What have I done? From Pedro. And Pedro, you actually, I think, had a couple of questions. If we can get one of our team maybe to allow or invite Pedro to unmute, he could ask them himself. Meanwhile, I will read the first one, which is, why was it supposed that the mother of all lattices would have more connections per node?
That's a super question. For two reasons. One, because it's like the general evolutionary method of throwing away unnecessary stuff. We can see it anywhere you poke, like take the brain connectome, right? We start with an overconnected brain, and then around mid-childhood, it goes through pruning, right? So in evolution in general, there is a surplus of organisms that get selected naturally or sexually, and only those that by virtue of variation and just random fit into the circumstances, they carry on and procreate. And in bone specifically, there are some seminal works by the lab of Tim Ryan on embryonic development and evolution. And they found, using also microcomputer tomography, that in early bone, in utero, in embryonic bone, there is, it's like super connected. It has very dense, very uniform, very fine struts that are not even on the scale of a mature bone interconnected strut network. It's much finer. But the point is the number of connections by far exceeds what we have in our mature skeletons. And then with the onset, after birth and with like crawling and the first attempts to locomote, certain elements get discarded. They just disappear by bone remodeling. And the end result is more sparse and more meaningful and coarse trabeculae that are connected less than it was in the original template from which we start. And we can actually see that not only in humans, but in all animals. So the primary bone is very fine, it's very uniform, and it's awfully interconnected. So we decided to mimic this natural process of pruning because we just thought that it's easier to discard unnecessary stuff rather than to build new elements, because we didn't have any assumption where to build and how and what would be the premise. So we decided to go from the surplus side.
So thanks, Natalie. Pedro, if you have a further question, but on this same topic, I want to just take it another step. And I'm really curious to know if you went on to generation 5 and took away even more. Many of the people here are therapists of some sort. Would that then lead to instability and such?
Yeah, so that was exactly that inflection point in the third graph. I think that was also Pedro who asked that the next question I see in the chat box. Like, what does that inflection? So imagine you keep discarding elements, element by element, and nothing changes. Your structure is as rigid and reliable, and all elements work in tension and compression. And you keep doing that over and over and over and over and over, and nothing changes until you get some critical point when you just have discarded too much, and all of a sudden, non-linearly, you end up with domination of bending stresses that are super effective in breaking anything. Any material is about 10 times less strong in bending. So bone as a natural lattice stops just at the limit of going into this dangerous mode of bending dominated loading. And so it's like a bargaining, how lightweight the structure can be before it enters the dangerous mode of operation. So it's the last reliable version of evolved and lightweight assembly.
And then we see Hila's images of the evolution. And am I correct in understanding the body or throughout evolution, we're adapting to what basically is put upon us so that we can survive as best. And it's how do I put this? Whatever we're asked to do, that's what is brought forward. Okay, now we have a question here from Patricia Hopper. Patricia, if you want to unmute and comment further, you may. In the meantime, I will read your question. And Patricia writes, can you define the term stretch dominated as it describes to the qualities of bone you've described?
Okay, let me use a visual prop.
Thank you. Hang on, let a moment, Gwen or somebody, will you please spotlight Natalie so that she's big on the screen?
Yeah, okay. So I'm using visual prop. So I have a very simple lattice that is not three-dimensional, and it has four elements that are connected into a square-shaped assembly, right? If we load it from any direction that is different from exactly aligned with the vertical and horizontal struts, in any other direction, we will have bending in the struts and shear in the nodes. By adding just one element going diagonally, we triangulate the structure. And so now, I don't have enough fingers. So imagine I hold it like that, and now we have 5 elements. So it's like an investment of 20% more of material, but now everything will change dramatically, because if you load this structure in any arbitrary way, for the ease of handling, and now I will show the triangle, right? If you load it from top to bottom, then the top 2 yellow pencils will be loaded in compression and the green pencil will be loaded in tension. And then in any other way, you will have loading along the axis. And anything loaded along the axis, either in compression or in tension, will perform better than the same object performing in bending. I don't want to ruin the pencil, but you can picture what will happen if I exert too much of bending force on it. So without changing material, without changing the amount of elements by much, by connecting them in a certain way, you may achieve higher stability by triangulation, or you can actually, if your purpose is to design a sacrificial structure that will crumble upon loading, that's a good goal for certain applications, right? And then you may favor easily deformable rectangular assemblies. So that's why I call it like dangerous, because we really don't want to experience any plastic deformation in the trabecular bone tissue, because then it demands healing and recovery and the limited locomotion. So the assumption is we want bones being reliable at the least metabolic cost. So that's my interpretation of the results. I hope this answers the.
It does for me, and maybe Patricia can come on if she wishes. And I'm just going to reiterate what I'm understanding. Please correct me and direct me if I'm off here. But the triangulation offers a separation of compression into some elements and tension into others, which then means that we avoid these, or at least avoid the bending moments. And then I don't know if you've considered or not, in the biotensegrity paradigm, we're also considering each of this triangulation or the tensegrity architecture down in multiple scales. So if it appears to be bending, and this is within the constraints of the material, on one scale, when we go down a scale and more scales, it's just the redistribution of the forces.
Right.
Okay, great. Patricia, you good? Okay, and I'm going to go on then and ask George's question, if George is there and would like to comment. Please, I welcome you to unmute. So George Roth asks, how can these concepts be transferred to the properties of cortical bone?
Not so easily, because cortical bone, actually, can I show 2 slides? I have
Yes.
2 spare slides, and it's from the times when Hila and I worked at the Weizmann Institute, and we were studying, well, bones, obviously, and then we had a really puzzling experiment. So there was another student of the group who was a dentist, and he wanted to explore the behavior of trabecular bone in in an available animal model, and he knew that in the human mandible there's a huge amount of trabecular bone. You just open the cortex and you can harvest as much as you can. But you wanted more than that than you can get from a human. And the plan was to get trabecular bone from the mandible of a pig. Get a pig, cut the mandible, harvest trabecular bone, and test its connectivity and mechanical behavior and analyze it by imaging. So we got the pig mandible, which is like really huge. It's of the size of my forearm. And then we cut it at the middle to put it into the scanner because it was too big to go in its entirety. And we were shocked to find that it was hollow. It was like a human femur with a dense cortical bone. And there was no trabecular bone inside. There were some teeth sitting at the top with the roots, and that was it. So that was a huge disappointment. Why would you have a femur in your mouth? What's the role? Because such a hollow tube structure is the engineers know is the best adaptation for bending, right? If you offset all the available material away from the central axis, such a pipe will withstand bending in random directions with the best efficacy. And there's a formula for that. But then the solution came when, I couldn't sleep, and I started reading about what pigs do. And actually they did. They used their mandible with the incisors as a trowel to, search truffles or pick tubers from the ground. So they exert on their mandible enormous bending force. And the best adaptation for that is to install this tubular hollow structure in the body of the mandible. So they have no reason to fill the interior of the jaw with trabecular bone because it doesn't work this way. So cortical bone in vast amounts can be found in the hollow shafts that operate in bending because muscle attachment happens in just predictable positions along the bone. So it looks like bone is bone, right? And it's the same material everywhere, more or less. But in fact, these are different structures. It's like from the same brick, you can build a bridge and a house. So that's along the same line.
So if I can just approach that, thanks for the answer. But one of the things that I've heard you mention before, Stephen, is that long bones in the living state are very different from what we observe in the dry models that we often see. And really it's a collagen framework, right? Essentially mineralized. So, living bone, from what I've heard before, you can take a long bone like a femur or tibia, has about a 30-degree angle of bending that you can apply to it externally. And so, the properties here are such that in the living structure, the translation of the trabecular properties to the cortical bone, there's a framework here that has to be continuous. And in my experience, this is the main shock absorbing element in loading and, compression and so on. Yeah. So to me, just stopping at the trabecular bone is a little frustrating for me because in my work, I've seen cortical bone dramatically respond to the application of therapy to the point where I describe it as a non-linear process. We have our practitioners using calipers and tape measures, and in one treatment, they can change the shape of the bone. And that's including the shaft, the cortical, which is mainly cortical in the especially the weight-bearing bones. But yes, you have a comment on that?
Yeah, I want to say thank you for your perspective on this. It's exactly right. So I watched a few talks on human biomechanics, and somebody made a comparison that when Usain Bolt is sprinting, the top of his tibia bends by about more than an inch with respect to the foot. And that is incomprehensible because we used to think of bones as rigid bodies that are like, skull and bones, white and not so dynamic. So it just puts another dimension and another level of complexity that we have really to work hard to get to the correct interpretation of what we know. So thank you for this. And it's really humbling because we can't really say that, okay, this is the truth. We have to look at the whole picture as Hila highlighted.
Yeah, and I think one perspective might be within cortical bone, it's not such an obvious, space frame as within the trabecular bone, but it still is a space frame. It's not totally dense, and it has. But if you have to look at the collagen framework and everything else, those elements actually have a tremendous amount of tensile bending and compression loading properties that are very similar to trabecular bone, and each one serves its purpose for different parts of the overall functionality, I would assume. Yeah, so that's great. I really appreciated your talk, and I think it helps me understand the extrapolation of the engineering and physical properties of bone and how that translates into structure. To me, structure and function are intertwined, and so this adds to that discussion tremendously. Thank you.
Okay, thank you. We've got 10 more minutes until you need to depart, Natalie, and 2 more questions for you. You've already addressed Pedro's. Okay, so this is from Elizabeth Larkham. Again, Elizabeth, if you'd like to unmute and pop on, you're more than welcome to do so. Do you apply your research to prosthetic design? I thought I heard the mention in the introduction.
Not really. I had some ideas that could be applied, but then it's just, I think we're still not there. We still don't understand the complexity to kind of drive it into the field of regenerative medicine or prosthetics or robotics.
Oh. Can I add one point to that? Because I have had experience with this, with people who've had prosthetic joints. And what seems to happen is there's a failure after about 6 months in certain cases where there's severe trauma. They use a glue to bind the prosthetic, but that usually only lasts for about 6 months. Then they expect the bone to grow into the finer components within the prosthetic. So what we have had several times now where this failure occurs and we apply therapy to the bone to restore some of those natural properties after the injury, restores that flexibility, it actually rapidly stabilizes to the prosthetic. So we've had cases where people were scheduled for revision surgeries. We've been able to avoid them in at least 2 documented cases with that. So I think there is direct application here, but it needs further research for sure. Okay.
Well, that's exciting news for anybody in that situation. So we have, Pedro asked a question about the last graph, which you have answered, and Mariana Barreto, if you're here, you can unmute if you wish. She's asking, are these measurements or simulations done in live or in vivo as well?
No, these measurements that I presented are all in silico. There's our simulation measurements because we are talking about the graft, the skeleton of bone, if I may call it this way. So we kind of undress all the trabecular elements and we only keep their centroids. You can't do that on the real specimen because how? There are too many of them and they're too tiny. So we extract this information about the graph of bone, which is the abstract mathematical representation of a 3D body. And then on that dimensionless grid or lattice of edges and nodes that don't have size, they just have the coordinates of origin and end, then we can apply simulated loading. And then, of course, you can compensate for bending dominated behavior by adding more material, which is the case in real life. But any, even a thicker structure would be in a state of disadvantage if it is loaded and bending. And you can save some material of metabolic expenses for maintenance of bone if that element is loaded in tension or compression. It's just metabolically cheaper. So that's why we kind of try to ignore the real life size and shape of elements. So it's mathematical.
Thanks, Natalie. Mariana, any comments? Okay.
I'm just having a hard time. Sorry.
Go ahead.
Because in my head now, I think we learned in this community to think about soft matter and to think about bone being flexible and behaving differently when we are alive. So, I mean, I found fabulous what you presented, but now I'm having more doubts in my head. So I guess, thank you. But now I have to learn more. Thank you.
Thanks, Mariana. Now, Natalie, I know you need to go shortly. I've got one question for you, and then I hope that Steve may hop on and say some final words before you need to depart. And this is not directly about your presentation. We talked a little bit about it earlier when we spoke a couple of weeks ago. You're at McGill. You're an assistant professor there. One of the discussions that we will frequently have in our community is how do we forward biotensegrity in different disciplines, in different communities. And I wonder if you have some comments about your thoughts. How do we forward biotensegrity in academic circles?
By infiltration. We have this established paradigm of classic mechanics, right, and classic tools. And we tend to think along the established lines, established paradigms. So there is no way to overturn the common concepts in materials and physiology, anatomy, and biomechanics. So just by tiny increments, step by step, we can introduce and just making people interested. And maybe it's not up to a single enthusiast to come up with the necessary critical mass of disruptive ideas and good solutions. Like, as it takes a village to raise a child, it takes a department or university to start a course on tensegrity and start teaching it in a systematic way with the proper toolkit for experimentation, and then it will spread. So it's like, it will take time.
I know you said, I wrote it down because when I asked you that question earlier, you said evolution, not revolution.
But yeah, that's right. Thank you for reminding. That would be my approach.
Yeah, no, you put it so beautifully and in such a memorable way. Steve, would you like to pop on and share some thoughts?
I'm muting myself. Well, of course, it was a exciting presentation, and I think some of the questions that Mariana brought up are certainly germane. And what you just brought up about integrating into the formalized biomechanics population is very important. And actually, I heard the talk given by their Huskies in 2000. So I've been working with the same problem now on bone since 2000, when I discussed it with him directly, because we sat together at a meeting for 3 days and talked about it. And there were 3 bone researchers there who are now, well, 2 Huskies died, but the other 2 are alive, and both of them run labs, in the one in Australia, the last I heard, the other one in Germany, the last I heard. And this problem still arises. They still haven't integrated the concept of the micro and the macro. And until they understand that there's a range that you have to understand the macro and the micro at the same time, that you can't really understand what you're doing at either level.
Which is one of the beautiful things about Natalie's paper, which I read or tried to read in preparation for this. But watching the presentation, Natalie, makes it much easier for someone like me who doesn't have a background reading research papers. So I really appreciate that. Okay, so I'm going to keep you on time here. This is one of my goals as I host this and the things on the Embodied Biotensegrity platform. And everybody, Natalie has to run off in 2 minutes. So if you want to say final words to Natalie.
Or are we going to do closing toast before Natalie leaves?
Well, we could, but there are a couple of questions for Hila here. And
Hila, do you have more time?
I have more time. I'm happy to give a closing toast before Natalie leaves and then continue to talk about more questions.
okay, great. And we can maybe keep the YouTube on even after the toast so that Hila can respond on YouTube. Is that all right?
Yeah.
Great.
Okay. Thanks, Susan and Hila.
I'm going to bring this back up just because I think it's funny. But
Great.
speaking of cellular solids, Natalie. So I am pretty bad at giving super formal toasts, but I want to start by firstly just thanking the entire team here, the Biotensegrity crowd, and not just for the Biotensegrity parties, but the course that was run, and really just providing an incredible, welcoming, and really deeply inclusive community to discuss all these things with. Like, it is really rare for me to find spaces that are so accessible to such a range of perspectives and backgrounds, to the most hardcore detailed scientific research and total other artistic or cultural perspectives, and functionally being able to have collaborative and integrative discussions. It's an incredible space that you have all cultivated, and I really appreciate that. And I also want to really specifically thank Natalie for also cultivating a space like that, especially within me, and just giving me a way to experience being a scientist and a researcher in a really integrative and holistic way, where we can be talking like super hardcore math details about bone structure and topology, and then in the next hour be like rushing to put our goth makeup on and go out dancing. And it's just really wonderful. And I am just really excited about especially this community and this context of not just being good for discussion and interest sharing, but having really action focused. How do we get biotensegrity and these kinds of approaches into all these fields and into all these spaces, and how do we work together and coordinate to really do that? So I'm really excited to be a part of this. Salut.
Thank you, dear.
Salut. Thank you so much, Hila. Natalie, cheers.
Chin chin.
Okay, thank you, Natalie. That was amazing.
Natalie.
We'll see you again soon, I hope.
Bye.
And then, bye, thanks. And then Hila, first question for you here is from Pedro.
Okay. Hi, Pedro.
And Pedro, if you're here and you wish to unmute and add to this or ask the question yourself, just pop in and interrupt me. Meanwhile, because I don't see everybody on screen, I'm just going to start reading it. If I understood correct, the graph shows a turning point. Whoops, is this the right one? To Hila, yes. If I understand correct, the graph shows a turning point to a zone where super holds bending, but very low hold compression tension. Is that correct? I hope you're here, Pedro. Is Pedro here?
He's unmuted.
Okay, great. Why don't you ask your question, Pedro?
Yeah, go for it.
Maybe he's having trouble. I don't see the camera on, but I did see him unmute.
So if we're talking about an inflection point, I have a feeling that was a graph from Natalie's.
Pedro, we cannot hear you. He's asking in the chat. Can you
Okay.
hear me? No, we can't hear you and we can't see you. His camera won't turn on. Okay, well, we'll just remember what you look like, but it would be nice to hear you.
Hang on, let me try that.
Try again, Pedro. I think I heard you that time.
There he is.
Oh, I
We
see
see
him.
him. Yay.
Can't hear you.
I think it's a connection. Try one more time.
Either that or you're on the wrong mic. Oh, now you're muted. Now you're unmuted. If you take your cursor to the up arrow beside the mute bar and just check that you're on the right mic, that might help.
You know?
Yes.
No.
Yes.
So probably a problem with these, right?
Probably.
So first, thank you very much, Hila and Natalie. I think she's not here anymore. That question was about one of the last graphs that Natalie presented. She was pointing that she found out that the bone was located exactly at the turning point of something. And I wanted to hear more about exactly what the graph is about. What's the y-axis was not very clear.
Oh, sorry. It said, I read wrong. It said Hila here, so I misunderstood. Hila, do you want to take this or?
You can attempt. I'm going to try to remember what you're talking about first. Are we talking about a stress strain curve for bone material, or are you talking about the actual data from the study that she was presenting?
The ability to hold stress from bending or for compression, something about that.
Yeah. I mean, I can give you like I can like Google a basic bone material stress strain curve. Like I think.
But I
I'm
think
remembering.
that inflection point.
Yeah. Of where it goes from plastic to. Yeah, static formation. So your question is what the inflection point means?
Yeah, because after the inflection point, something went skyrocket, right?
Yeah. I'm like, I wish I had the graph in front of me, but
For
I'm
bending.
assuming.
It was for bending. She was talking about how the bone wasn't allowing bending, and then at one point it allowed bending. So yeah, that peak there was for bending. The other was for compression and tension.
Let me grab a graph, but I'm just gonna talk as I try to pull something up for you. But in general, so with.
Can I say one thing in the meantime? Sorry.
Yeah, go for it.
So we have to remember, right, that there's the action of bending, like I'm going to take this stick and I'm going to bend it. Okay. That's one way of using the word bending, right? But then there's the bending moment, which is more the physics term, right? And when you have a tensegrity mast, it does not, remember Natalie kept saying it does not allow bending. In other words, the tensegrity structure at multiple scale levels, you can make that motion of taking the ends of the tensegrity mast and making it shape bend, to make it go into a more rainbow shape. But what won't happen, because of the balance of tension, compression forces all down through the system, is you won't have within it that bending moment where within one piece of material there's a fight between push and pull where things will snap. Am I on the right track, Hila?
Yeah, no, that's a wonderful explanation, and actually really gets at like why I care so much often about being really specific and detailed about what the structure actually is, or like what we're talking about. Because when you look at matrix structure, you're looking at trabecular bone. Even if you're considering it totally separate from the whole bone or cortical bone, you're still in this situation of you have you have something made up of many elements and in a configuration. So the whole thing can have a behavior or a function. Each part of it can have a behavior or function. Technically, you can go beyond the trabecular strut level and look at like the more cellular material or the hydroxyapatite and collagen interface. To get really quickly at just what I think you were talking about, Pedro, like this, when you're just looking at the behavior of things under loading or in functional context, it's just kind of describing that there will be a point for some materials. So for bone, it does have a plastic deformation region. It's not, so you can look at stress here, you can look at displacement or strain, but there's a very small window, I guess, if you're putting a bone under loading and you're finding this. But the thing I'll point out too is often these kinds of, like Susan's explanation just now, does a lot better of a job in an integrative context of thinking about how things might be working and moving and you know, sort of in situ. Because a lot of the time when we take material, especially for bone, like you can look across papers and meta studies of what the stress strain curve for bone is. And it really depends how you measure it. Are you measuring a whole bone that is wet still from a live organism? Are you measuring a museum specimen or a dried bone? Are you measuring a piece of bone? What kind of bone? What's its shape? Is it in 2-point bending, in 3-point bending? So when you do these too simplistic sort of material properties or rules that we talk about, it can be really hard to use those results to integrate into a fully holistic understanding of what's actually happening. Even with just when something might go into plastic deformation and then break, but no longer be super rigid.
Can I try something
Hila,
else?
I was gonna say, Hila, I've taken a screenshot of that graph from the YouTube. Would you like me to put it up on screen?
Of Natalie's graph?
Yeah, I just went back and
sure.
Yeah. Then we know Pedro can tell us if we're on the right page.
I'm a very visual person, so
But
it's like
either way, whether it's the right graph or the wrong one, I want to follow up in a minute.
Okay, well, why don't you go ahead while I'm putting up, well, Hila can decide what Hila wants to do first. How about that?
Okay, I mean, I doing my best to answer your questions about Natalie's stuff.
Yeah.
Okay, yeah, so this is all computational, so that's another interesting element here. But so you're talking about this sort of inflection point or point where it shoots up in silico. Yeah, exactly. Yeah, okay. My thoughts about in silico are interesting because it's like, it really, if you're computationally modeling something in only digital space, it will have differences if you do the theoretically exact same test in actual material 3D space, but both technically count. So your question about this graph being where it shoots up in stress modulus, is that
your question, Pedro?
That is my question, but I see that perhaps we should
well,
ask
so
Natalie,
this
not
the
Hila, right? And
the x-axis here is the coefficient of decay, which we actually were talking about a couple weeks ago when Natalie and I were talking about our talks and stuff and like integrating things. And so I think, so the connectedness decay coefficient is kind of from, like, I can't explain things as well as she can probably, but like it is a bit of a fancy way of just saying it's a metric for how many like elements there are, the connectivity, because we're talking about topology, so this overall format. So we don't care how thick or thin anything is, or there's only straight lines technically. And so what it's saying is that with these iterative generations through pruning and then forming more connections, and really importantly, this is done computationally within bending. So you have that sort of like evolutionary force, or that sort of feedback force that the algorithm's considering to change the structure. And so as that increases, the connectivity coefficient, so just this sort of structural measurement or metric, what you're seeing is the actual bone, which has a result, like it has a certain amount of connectedness has a certain topology, and that's around, let's say, -1.5. Not super important what the numbers are, but she was just showing that through iterative generations computationally, it approached the very realistic results that you get from the bone matrix itself. So you're proving the concept that this is computationally kind of AI getting toward a sort of evolutionary process or developmental process that gives us pretty similar properties within this metric to real bone.
And
would it be, say,
can we keep it up? So when it says contribution of bending, is that what I'm understanding? So tell me if I'm wrong here, is that through the computational process, we are applying an action as if we were trying to bend the material.
Yeah, so I had a long discussion with her about this because like I just have pet peeves about only digital computational modeling, but she said that that the algorithm effectively considers them in bending. I mean, one was definitely straightforward compression of these cubes, and then there's some way that it was put into a context of bending.
Yeah. So this is what I'm reading. Tell me if I've got this wrong, because I'm thinking of this is helping us maybe see how you break an arm. You know, when I was a kid, I broke my arm. I mean, I fell and I broke my arm, and it's like, oh no, you know, and at the humerus, it was in 3 pieces. Okay. That's a big bone, right? To break. Later on, you know, 50 years later, I say, oh, my arm broke to save my neck. It was a good thing for the humerus to let go.
Yeah, absolutely.
And so this is the point. You see what I mean?
Yeah.
Like it can take it. And when it knows it can't, and it would otherwise have to pass it up through the system, it would rather break to save something that's more essential.
Yeah. Can I actually riff off of that real quick?
Yeah, please.
Okay, because that is one of my, so a lot of, I'm gonna pop one of my slides back up real quick. So
Pedro, look at the trouble you've got.
So a lot of the reason I'm looking at birds, like aside from being really obsessed with flight in general, but like coming from the mammal and human world, birds are really incredible because they're really this pushing to the limit, the lightweight structure, this, you know, bone is cheap as long as your configuration is the right way, it can achieve incredible things. And this right here is this 1992 study where, so there's just been a surprisingly small amount of like research into the role of trabecular bone directly and mechanistically, and how that appears across the phylogeny. And so in birds, for instance, you have actual results, and I've been sort of playing around with this in other species, but you have a really nice study showing that the contribution of trabecular bone just existing in this bird humerus, so just like your humerus, Susan, not so different. There's a lot of interesting results with stress and strain locally and deflection, but the key result was that it directs fracture toward a midshaft. And when you think evolutionarily, similarly to, you know, sometimes things should fail because in an integrated system, there are different kinds of fail-safes and different kinds of contexts, because evolutionarily, you do have some pretty straightforward parameters that you have to like survive and reproduce. And so like the organism's life is going to be important. So that happens. So similarly, at this localized level with bone function, if there's an evolutionary drive, potentially based on trabecular bone, but if there's an evolutionary drive to have your bone fracture at a midshaft, not at a joint, it's going to be way better. Because if your joint morphology screws up, then you're not going to be able to move maybe the whole limb, maybe like that's going to affect your behavior, your life way more. But I've sat in the AMNH collections, measuring every single primate humerus and femur, and multiple had evidence of it was like completely healed, but you'd see the femur humerus have a break mid shaft, so it would go down, across, and down like a freaking L shape in the middle of the shaft, and it healed, and the animals seemed to continue living for a long time because, you know, maybe you won't, maybe it'll impact you a bit, or maybe your bone will be more or less robust to breaking there, but you're not going to mess up the way that you can move at every joint and your actual kinetics.
Right. And it did. It broke midway in 3 pieces.
Which, Hila, I think what you're talking about is this perfect example of taking it back into the bigger context that you've been talking about, because this was going to be my question for you, was to give us some examples of the roots-based integrative approach, and how do we then take this science, which you've basically just done, and expand it out to biology and back again. So I'll add one more question to that, and that is, I know you work with people of all ages, so how would you take something that's quite complex and present it in a practical, embodied way to somebody who's either younger or who doesn't have this foundation of knowledge that you have?
That was a great question. I mean, I will say this, all the methods that I find work best with adults are the ones that work best with small kids. So I'm rarely thinking about how to communicate things to adults and then trying to like figure out how to simplify it. I'm actually usually doing the opposite, where the framework is embodiment usually. So being able to have people understand their own form and function, move things, look deeper. And then honestly, I don't actually think that things are that complicated. So one of the things we teach in the art and science course, the roots-based integration, is like complexity is not the same as complicated. So it really matters how you present complex things. And sometimes it's just a matter of you have many different elements. So maybe 5 will fit into one session or one program. It depends on the context, right? For outreach or a course that's a whole semester or whatever it is. But the main thing that works with kids, and thus also with adults, is this hands-on approach and this, sort of direct examples, but not giving examples in a way that makes it seem like they're hard rules. Always trying to bring things into a comparative approach, always trying to empower whoever I'm working with to like see what's happening and think about the how, but not think, okay, that is exactly what's happening within this system, and not having the ability to apply the same concepts to other ones comparatively. So, I mean, one example, I can pull up one of the slides again if you want.
And this brings me to the question I wanted to ask before, because it's a little about teaching too, Hila. So it's a metaphor thing. So if I was to say, go back to that idea of bending versus a bending moment, right? So if I have a sponge and I bend a sponge, and there's foam being bent. There, okay, so I can think of that, and that's not directing bending moments because of the nature of the spongy structure. Now think of go beyond that. Think of a living sponge, right? Or the bubbles in the beer that Steve talks about, right? And where the way bubbles work, if you had some magic bubbles that were living, that could reconfigure the way the bubbles in the beer reconfigure as you took a bar, a stick made out of spongy bubbly stuff, and you could do this, and when you did, all the little connect between the node lines could change and shift, right? And the bubble could change, right? And then they could change back. Am I starting to get to the right place where this is where the structure doesn't allow the bending moment, as they call it?
I think that I mean, what you're saying is exciting to me, where like, so one of the things we were working on doing before I ended up leaving Weizen was like literally we have a really nice CT there where you can actually do a loading cell inside of it and image the same chunk of bone fully at like 5-micron resolution and then compress it and image it again. But what we were working on was more, again, on the methodology side, the computer vision and image correlation and all this mathy computer stuff that you need to go in at that detail digitally to look and measure each element. But it's interesting when you talk about the bubbles. I guess I think of, so like foams or cellular solids are generated through bubbles like traditionally, and part of the fun stuff with trabecular bone is different models from a fabrication and design perspective of like, well, with bubbles you're kind of trapped in to an extent at least into a structure, a topology that tends to be a little bit more hexagonal, or tends to have like, it's going to have a constraint because bubbles are spheres, right? And so it's not the same as a matrix that's forming in a different way, that's not forming from the negative space of bubbles left behind. And so there's just a whole lot of different kinds of ways to think of cellular solids, but again, it's very much influenced by how we think of these things in our like human systems, like bread or airplane design. I'm not sure.
But just as you're going to, if you want to draw like Voronoi patterns, right? And you start by just putting a bunch of dots on a piece of paper, and then you do the circles, you know, you find all the different circles and stuff. Somewhere between that and what happens when you try to draw like an Apollonian gasket or something, where you've got, you know, the bubbles within the bubbles within the bubbles. Something to me is connecting back to what we see with Jean-Claude Guimberteau's fascial reconfigurations, where the sort of densifications and the alliances change and get created. I don't know if Steve has.
I can try to respond to that. I'm not sure where we are in the question answer. I want to like visualize that more.
I love what you're talking about and how you help us go back to the whole root space thing is fabulous, right? Fabulous. It's like, why are you wasting your time trying to, you know, and the image you use, you know, trying to build a connection from this tree to this tree. They're already connected. It's really wonderful.
Thanks.
So I will step back and let you talk.
Oh no, you're. I'm just trying to like. There's so much. One of the hardest parts about being deeply integrative is it's like finding the way to like make things make sense in infinite complexity, without needing to bounce around from one thing to the next. Like what are like, that's the helpful thing about models. Like we're necessarily locked into thinking in terms of models, but we just need to know how to use them. But like I would go past that a little bit often, where if you're thinking about something, you can just think of the thing actually and not always use like a top-down term. But if you're going to, just knowing where things exist in this sort of network or web of reality is helpful. But it's complicated to talk about because you bounce around and it's like, oh, that's related and that's related. I just have a slide up, so I'm gonna just quickly answer part of, I think, Chris's question is like giving a really specific example. One that I love giving is, so I have mushroom anecdote written here.
So before you do that, Hila, can you expand so we see your whole slide?
Yeah, let me see if I can.
Right now we see your.
There we go. Okay.
That's great.
So of course there's movement-based ones. There's fun activities. There's so many different activities that I've run, and a big part of what I'm doing now and have been for the past year is like helping. So I do a lot of workshops to help people develop their own integrative activities for teaching or also for performance, like full-scale arts performance or galleries. But I like pedagogy and children's museums and things like that. And so there's a lot of fun movement-based ones, but one of my favorite ones to run is like a crafty one where you're just using, so you can use marshmallows and toothpicks, right? And I feel like a lot of folks that have been into biotensegrity or things have probably tried something like that. And you can look at different configurations, talk about the triangles and the orthogonal stuff that Natalie was mentioning, and the tensegrity structures that are built on themselves. But this one is more evolutionary focused. So instead of marshmallows, there's clay. So you have more degrees of freedom for the shapes that can be created by children, not just marshmallows. And the point is making body forms. And part of this is a paleontology exercise, because when we think of behavior, we think of movement, we're also thinking of environment. We don't move in isolation. We always move in up against and within our environment. And that's just as much a factor of how we move and what we do as our bodies or bones or any of the stuff we've been talking about today. And it's something that I harp on a lot, because people think of birds and flight as like not in an environment, it's just an empty space. It's like, no, like it's just air and we can't see it, but it's just as much of an environment as water or terrain, ground, rock, sand, etc. So here, basically they get to design creatures however they want, with however many limbs, however many body length variations. Sometimes they give them cute ears, but then they take them and paint and they stamp and they make footprints. And there you can also talk about how fast, or if they're moving the legs, or if they're hopping, or like all these different kinds of locomotion. And then you have them trade and try to match up the footprints to the creatures based on what they look like. And you get to also talk about how it's hard sometimes if you don't see them in motion to match them to the footprints versus if you do. And my favorite anecdote is that one time I ran this activity and a kid who was like 4 maybe was just sitting there and he sculpted this beautiful little guy. It's not pictured here, unfortunately, but it's just like one toothpick and this like thing and a head of some sort. I wasn't sure what it was. And then he sat there, and when everyone else like did their footprints, he didn't. And then everyone was trading their papers and creatures, and he like was going to, and I was like, hey, like what's the matter? Are you, like, I thought he was uncomfortable, and he's like, oh, mine is like a mushroom. And I was like, okay. And he's like, yeah, it doesn't move. Like it doesn't locomote. And I was like, oh, cool, very valid. That's a valid paper with no footprints to connect to an organism's structure, function, reality. So it's like, it's amazing, because that is the kind of thinking that I look for in kids, or when I'm teaching anyone, of like really obviously comprehending the entire point of an exercise and going further, or being personal and creative with it, making those connections themselves.
That's a great story. I'm looking at the time. We're at 1:30. So if it's all right with you guys, what we'll do is a quick looking ahead, a quick sponsors, and if you are able to stay on for the after party, there are a couple more questions, but again, we don't want to impose ourselves upon you, so it's totally up to you, Hila. Susan, are you able to do the looking ahead for us? Maybe I'm going to read the sponsors while you get ready to do that. How does that?
Thanks.
Okay, so a really quick word from our sponsors. We have Handspring Publishing in Scotland, Embodied Biotensegrity, which is here in Canada with Gwen and I, the Second World Fascia Congress. They'll be running an event in August. Fabiana was here earlier. I'm not sure if she's still here or not. The Fascia Hub in the UK. They produce seminars, fascia-related topics, integrated biotensegrity with Lisa Babiak, who may still be here. Matrix repatterning, that's with George Roth. We heard from George today. Artifact Pro in Madrid, Spain, and Pretense in Holland. Also Bruce Hamilton, who's in California. These past 3 are creating fabulous tensegrity structures and all 3 of them, I think, have them available for sale. And finally, for those of you who are with us on Zoom, you can find details of their discounts in the Zoom chat. If you are with us on YouTube, you can always sign up for the Biotensegrity Archive newsletter, which goes out on Wednesdays, and register and join us on Zoom, because we always love to meet you. Are you ready, Susan?
Yeah, I think so.
No, next week.
Next week, Neil Galloway's been working on connecting his understanding of biotensegrity with a book called Microcosmos, not to be confused with a lovely movie on bugs called Microcosmos. This is the work of Dr. Lynn Margulis and her son Dorian Sagan, and they wrote a book about it and covering her work, and it's some revolutionary evolutionary stuff. Neil's been connecting that with his understanding of biotensegrity, and he's going to be presenting on that, origins, evolution, and biotensegrity. And I'm excited because it's some very cool stuff. And Lynn Margulis Margulis, I keep saying her name wrong, was at UMass Amherst. My daughter, when she was at Hampshire College, worked closely with a friend of hers, another Lynn. The other Lynn, who was the scientist in the Valley, was my daughter's mentor when she was learning how to clone genes. So we've been following her work as a family for a while. So very cool stuff.
Okay, super. So let's say goodbye to everybody on YouTube. Bye, everyone. Gwen